Hello everyone, I am Doménica Pinto and I'm going to talk about exome sequencing and the management of neurometabolic disorders. First, we're going to start with some definitions. What are neurometabolic disorders? These are the result of an alteration in the genetic code that expresses enzymes, which causes their malfunction, affecting the development or function of the nervous system. It occurs mostly since childhood, but it could also be present during adulthood. And examples of these are ataxias, movement disorders, childhood epilepsies, and peripheral neuropathy, etc. Now we have a video showing a professional in the field of neurometabolic disorders talking about why he chose to study this field. Let's watch.
The reason for rare neurometabolic diseases or inborn errors of metabolism with severe neurological component is that because when we founded Minorix, our intention was to focus on rare diseases. In the very origin of the company, this is to provide new therapies for rare diseases from a broad concept point of view. But there are many rare diseases, several thousands. No? So when we started to fine-tune what we are going to do there, we decided to focus on inborn errors of metabolism or rare metabolic diseases, because these are a group of diseases which are very severe, where there's a very highly unmet medical need, because in 90% of the cases there is no therapy. This is similar to other rare diseases, actually, this ratio, but diseases that affect mainly or most severely children, because are genetic disease, so the basis of this is genetic, so that's why affect also severely children. But the main reason was because technologically we believe that was the kind of disease where we could provide a kind of solution, at least for some of them. And that's the main reason at the end of the day was technological. And what are they? These are a group of, as I said, genetic disease that affect metabolism. This means that there's one gene that has a mutation, and because of that mutation there's a particular protein that is not working well, and because of that there's a metabolic process that is not working. A metabolic process means either the process of where within the cell a big protein, for instance, is cut into pieces, or the process where a big protein is built into pieces. So these are the kind of processes that are very relevant in the cell. And when this kind of process is not working well, you start to have different kind of problems, and cellular dysfunction, and at the end, cell death, or consequences in different organs, such as neurological consequences, and so on and so forth.
Now, what is exome sequencing? Exome sequencing is a technique that allows to create a sequence of all the expressed genes in the genome, which are called exomes. Now, we have a video showing us how the human genome is coded, what is the process, and after this we can watch a video about what is an exome.
You've probably heard of the human genome, the huge collection of genes inside each and every one of your cells. You probably also know that we've sequenced the human genome. But what does that actually mean? How do you sequence someone's genome? Let's back up a bit. What is a genome? Well, a genome is all the genes, plus some extra, that make up an organism. Genes are made up of DNA. And DNA is made up of long paired strands of A's, T's, C's, and G's. Your genome is the code that your cells use to know how to behave. Cells interacting together make tissues. Tissues cooperating with each other make organs. Organs cooperating with each other make an organism. You. So, you are who you are in large part because of your genome. The first human genome was sequenced 10 years ago and was no easy task. It took 2 decades to complete, required the effort of hundreds of scientists across dozens of countries and cost over $3 billion. But someday, very soon, it will be possible to know the sequence of letters that make up your own personal genome all in a matter of minutes, and for less than the cost of a pretty nice birthday present. How is that possible? Let's take a closer look. Knowing the sequence of the billions of letters that make up your genome is the goal of genome sequencing. A genome is both really, really big and very, very small. The individual letters of DNA, the A's, T's, G's, and C's, are only 8 or 10 atoms wide, and they're all packed together into a clump, like a ball of yarn. So, to get all that information out of that tiny space, scientists first have to break the long string of DNA down into smaller pieces. Each of these pieces is then separated in space and sequenced individually. But how? It's helpful to remember that DNA binds to other DNA if the sequences are the exact opposite of each other. A's bind to T's, and T's bind to A's. G's bind to C's, and C's to G's. If the A-T-G-C sequence of two pieces of DNA are exact opposites, they stick together. Because the genome pieces are so very small, we need some way to increase the signal we can detect from each of the individual letters. In the most common method, scientists use enzymes to make thousands of copies of each genome piece. So we now have thousands of replicas of each of the genome pieces, all with the same sequence of A's, T's, G's, and C's. But we have to read them all somehow. To do this, we need to make a batch of special letters, each with a distinct color. A mixture of these special colored letters and enzymes are then added to the genome we're trying to read. At each spot on the genome, one of the special letters binds to its opposite letter. So we now have a double-stranded piece of DNA with a colorful spot at each letter. Scientists then take pictures of each snippet of genome. Seeing the order of the colors allows us to read the sequence. The sequences of each of these millions of pieces of DNA are stitched together using computer programs to create a complete sequence of the entire genome. This isn't the only way to read the letter sequences of pieces of DNA, but it's one of the most common. Of course, just reading the letters in the genome doesn't tell us much. It's kind of like looking through a book written in a language you don't speak. You can recognize all the letters, but still have no idea what's going on. So the next step is to decipher what the sequence means, how your genome and my genome are different. Interpreting the genes of the genome is the part scientists are still working on. While not every difference is consequential, the sum of these differences is responsible for differences in how we look, what we like, how we act, and even how likely we are to get sick or respond to specific medicines. Better understanding of how disparities between our genomes account for these differences is sure to change the way we think, not only about how doctors treat their patients, but also how we treat each other.
Hi, I'm Wendy Chung. I'm a clinical geneticist here at Columbia University. And then if you have questions, again, talk about this with your doctor.
This is a video about exome sequencing. In the past, we examined each of the 20,000 genes one by one for the genetic mutation that was the cause of the person's genetic disease. This took a long time. Now, we have the ability to look at almost all of our 20,000 genes at one time using powerful computers. This is called exome sequencing. An exon is the specific portion of the gene that we believe is most important for telling our body how to function and how to build working parts. This is where we believe most genetic mutations occur. The exome is the exons of all 20,000 of our genes. So instead of looking at all of your DNA, we can look at only the regions where we think most mutations occur. For instance, consider a time when you were reading a complicated textbook. Now, what if you could read only the important words or the concepts that you knew you would be tested on? It would take much less time to read, and you would be able to get the same important information. Similarly, with exome sequencing, we're able to read only the parts of your genetic information that we think are important or the parts that might have clues about your health. Who should consider exome sequencing? Individuals who have had extensive genetic testing, but a diagnosis has not yet been identified. Individuals who very likely have a genetic condition based upon family history, but the diagnosis is unclear. Individuals who have a genetic condition that could be caused by many different genes, and the cost of testing each gene individually is greater than the cost of exome sequencing. Individuals with an undiagnosed disorder who want to be as aggressive as possible about quickly determining if the cause is genetic.
Okay. What are the applications in medicine? This technique, exome sequencing, has an objective of finding a relationship between exome sequencing and the detailed study of clinical phenotype of each patient in order to identify new variants or abnormalities that which will be the causes of the disorders, and with this we could be able to find a possible cure. A study has been made and it had 41 participants, from which 28 had been diagnosed with 2 genes that had new variants that were unknown, 22 genes with new phenotypes, and 9 genes with expected variants that were already known and that had the scientists knew the correlation between the gene and the manifestation of the gene or the phenotype. What was the advantage of making this study? Well, it could be used in an 8-year-old boy that was involved in the study, who had autism, had movement disorder, intractable epileptic encephalopathy, and persistently abnormal neurotransmitter profiles. So he was a volunteer to had his genome coded, and the exome was also studied, and they found that he had a deletion of exome number 14, and with the study of his phenotype they could also find that he had abnormal sodium channels, and he had problems with transport of neurotransmitters. So this was a correlation that scientists they recorded this correlation, and the boy was given a treatment with catecholamines as levodopa, carbidopa, and 5-hydroxytryptophan and dopamine, which was very effective for him. Now we're gonna see a video with an introduction to these neurological disorders in children and how do they manifest in them.
Hi, I'm Dr. David Hill, and today we're going to be talking about a pretty broad topic, symptoms of neurological disorders in children. Now, this is kind of too broad to even get a piece of because, I mean, there are child neurologists who do a residency and a fellowship in diagnosing and treating neurologic disorders in children. That is anything having to do with the brain or the nerves anywhere in the body, so that's huge. What I'd like to do, given our limited time, is to just talk about a couple of the most common disorders and how you might recognize them. So first of all, and scariest, is seizures. Now, a general tonic-clonic seizure, what we call a grand mal seizure, is pretty obvious. The child is lying on the floor, shaking their arms and legs, their eyes may be rolled up in their head, their tongue may be deviated. It's pretty obvious. But the seizures that people aren't so sure about are called focal seizures or absence seizures. Those may just be staring spells. The hallmark of that type of seizure is that a child is not paying attention at a time when you think they would be or should be. Now, children stare off into space all the time. I stare off into space all the time. That's not unusual in a boring class. Maybe their parents giving them a boring lecture. It's dinner time. But when you get their attention, hey, look over here at me. Yo, right here. You expect them to look and check you out. Or maybe there's a loud noise or somebody else comes in the room. If a child is having one of these seizures, they don't look, they don't respond. Unresponsiveness is the hallmark of seizure activity. So if you see that your child is having a staring spell and not responding, that really needs to come to the doctor's attention. Now, another common thing that we see in children is headaches. That's a huge field, but let's talk about the headaches that bother us. First of all, don't assume that the headache is due to vision problems. Vision problems are rarely the cause of childhood headache. I'm not saying never, but it doesn't happen a lot. So yeah, I mean, you can get their eyes checked, but don't be surprised if their eyes are fine. Probably the most common cause of recurrent concerning headache in children is actually migraine. And look around the family for somebody else who has bad headaches. They may say they're sinus headaches, but they're probably migraines. Migraines do tend to run in families. We know now that even infants can have migraines, even though they can't tell you that's what's going on. And young children commonly get migraines. There are great treatments for migraines. So if your child is having chronic headaches that are stopping them from doing what they want to do on a recurrent basis, for gosh sake, get that child into a doctor, because the doctor can figure out something that should be able to make it better. Now, the headaches that bother us the very most are ones that wake children up from sleep, ones that cause vomiting, ones that are getting progressively worse, more severe, more common, ones that are associated with difficulty walking or talking or some other function of the body. Those all worry us tremendously, and those headaches all deserve immediate medical attention. Now, remember, stuff that's neurologic happens in the head, it also happens in other parts of the body. One of the things that always concerns us are alterations in gait. If a child is walking or running fine, and then for some reason suddenly is not, or suddenly they're walking on their toes, or they're arching their feet they didn't used to be, they have progressive weakness, there were things they used to be able to do that they can't do now, those things are always very concerning, always deserve a medical exam. There are dozens of other neurologic problems in children and dozens of other things to say about them, but for today we'll just talk about the migraines, we'll talk about things that could be brain tumors, we'll talk about the weakness, and we will talk about, what else did we talk about? All those things. So talking about symptoms of neurologic disease in your child, all of them, I'm Dr. David Hill.
Pediatric neurologists to care for kids with everything from migraines to autism to ADHD to seizures. In fact, Dr. Javier Valero says up to 5% of children will experience a seizure.
They lost awareness. They may just have a blank stare or some shaking of the extremities and be unresponsive for some minutes. One of the most common type of seizures that we see in children is febrile seizures. These seizures that are in toddlers caused by fever.
Parents should stay calm and make sure children don't hurt themselves during the seizure. After your child recovers, call your pediatrician. Seizures are very treatable.
We will offer medications, anti-seizure medications. We also have some type of diets that helps with seizures. We have rate of cure or controlled seizures up to 80 to 85%. And with new techniques of surgery, sometimes we reach 90% of cure or seizure control.
Now, what are the pros and cons of doing this exome sequencing? Between the pros we have, this method provides a new point of view for a diagnosis. It can reduce costs by discovering simpler treatments, like before they had complicated treatments that cost a lot of money, but now since they have found a specific cause they can treat it by just that cause. Who knows, maybe they can eradicate the problem forever and not having to have a chronic treatment. It also provides new criteria for researchers on the neurometabolic disorders area. Scientists, by the making of new studies, they have new information about this, about exome sequencing and its manifestation, which can be useful because in the near future this could be used as a basis for diagnosis. And in the cons we have, it requires detailed techniques to get accurate data analysis. Also, the correlation of genotype, phenotype takes a lot of time and patience. Patience, yes, because scientists haven't yet discovered all what the human genome means. So this will take a lot of time, and also it will take a lot of money and technology. And it's still a broad area of research that may not give expected results. If we are involved in a study like this, we might have our human genome coded, but we might not have the results that we expect. Like if we are involved and we have a disease, we might not get to be treated or not get the cause that we are looking for, because it's still in study. And about this issue applied in my country, Ecuador, we can say that no studies have been made yet, and there are no resources, like we have no economical help from entities like the government. And we also we have a lack of technology, and we have a lack of professionals in the genetic field. And geneticists, people who want to be geneticists, they cannot study here. They usually tend to fly to other countries in order to study what they want, and they work over there, so it's a bit hard to take all that technology over here. And also it is a problem because we would have an advantage of putting that mechanism and that technology over here, because we have distinct geographical areas of interest that had like a tendency of having neurometabolic disorders. So if we applied here, we could probably know which is the cause of having this tendency of people who have these or that neurometabolic disease, so or disorder. That would be an advantage. And as a conclusion, we can say that exome sequencing is a new approach of science to the diagnosis of metabolic disorders. Neurometabolic disorders can be treated easily with a defined etiology. Once we know the cause, it is easy to treat the disease. New studies have to be made in order to discover new alterations of known or unknown genes. And Ecuador is not as near as other countries to use these methods, sadly, but it could be used in a future. Well, that's it. Thank you for your attention, and goodbye.